All right, this is the recording for Biology 105. It is February 2nd of 2026. Let's start each week how we start each week, which is looking at the course schedule. Here we go, we're in week 3. We're gonna wrap up our week 2 material today, which is our chemistry review, PowerPoint, and then we'll get into biological macromolecules. So we're laying all the groundwork. We're gonna really get into talking about what makes up a protein and what makes up a fat and what makes up a carbohydrate and what makes up your nucleic acids, okay? In your lab section this week, you are doing a classic carbohydrate assay where you're gonna be making a standard curve. You're gonna be measuring the concentration of glucose in some beverages. This is gonna require you to do a bunch of dilutions and to use the spectrophotometers, the skills that we've hopefully sort of reviewed in the last week or two. Great. Any questions about where we are so far in the course schedule? Okay, cool. For this week's lab, standard issue stuff. All right, there's a protocol that you need to print and bring to your lab. There's a pre-lab video right here, okay, from Mark. And then when it's time to do your assignment, which will be generating a standard curve in Excel with a figure and then moving it into Word, putting a figure caption under it, putting the findings of your lab in the figure caption, and then exporting that as a PDF, just running you through the process. The whole process, I've recorded myself doing it. It's a 19-minute video. So once you have your data, you just sit down. Well, actually, the data's right here. Once you got this data, you just sit down and follow along in my video, and then you're good to go. All right? Cool. And then you upload that, and that's your submission for the lab this week. Otherwise, the things to be aware of are we're hitting the add drop deadline. Okay, that's today. So if you're not in SI yet and you can make this 12 to 12:50 window work, then go for it, enroll in SI. If you're at all concerned about your success in this course, the lecture exams are where people lose points because the lecture exams are the only thing where you're right here and the only information's in your brain and it's a multiple choice exam, right? I'm going to be showing May, who's running your SI, the exams, all right? So she's gonna know exactly what's on those exams and will be able to give you lots of help. I think she's also gonna be running extra study sessions that will be announced for everybody. But otherwise, you can come to office hours or make an appointment with me to talk about exam stuff. The exam's not until week 5, but that'll be here before you know it. All right, it's gonna cover everything from week 1 through week 4. The study guide's already posted. Any questions about that? If I make changes to the study guide, I will send an announcement out to everybody. Awesome. Then let's go ahead. I think that's everything I wanted to talk to you about. Let's go ahead at 9:06 and start the lecture. That's not too bad, 4 minutes. Okay. So at the end of Friday's lecture, let me turn the fan down a little bit. It's a little noisy. It just sounds less angry, I think. All right. At the end of last lecture, I brought up water, and I sort of tried to connect this whole, well, a gram of water is a cubic centimeter of water, and a cubic centimeter of water, all right, it takes 1 calorie to raise that by 1 degree Celsius. So all of these units have all been sort of set up to orbit around water, okay? Water's important. The specific heat of water is the definition of a calorie, right? It's how much energy it takes to raise 1 gram of a substance by 1 degree Celsius. It's 1 calorie for water, okay? So check this out. Iron, ethanol, these things, it takes less energy to raise them by 1 degree Celsius, okay? So a substance that has a higher specific heat can absorb more energy without changing its temperature. And what that really means is that a substance with a higher specific heat can sort of hold that energy more stably, right? It can hold it better. It can move that energy around. It can absorb a lot of energy without evaporating. It can release a lot of energy without becoming a solid. These are all the important things about specific heat. And for us on the planet and water, okay, global ocean currents serve to redistribute energy from equatorial regions to polar regions. This circulation, all right, means that globally we don't have as extreme of temperatures at high latitudes as you would expect without this redistribution of energy. It also means that terrestrial habitats that are closer to the water experience less extreme temperature changes. You see that by living here, especially if you came here from somewhere inland at all. Our day to night temperature fluctuations, they're kind of intense, but they're not that wild, all right? And our summer to winter temperature fluctuations are also buffered by that, by the specific heat of water. Now, why does water have such a high specific heat? Okay, it all comes down to the hydrogen bonds. Those hydrogen bonds, remember, that's the intermolecular interaction, okay, between water molecules, really between any very, very polar molecules. Remember, dipole-dipole interactions, right? A strong dipole-dipole interaction between molecules where hydrogen is one of the atoms. We call that a hydrogen bond. Here's the point. Hydrogen bonds are stable. That means that you can put a bunch of energy into these molecules without them moving around more. So when we're measuring the temperature of something, all we're measuring, what we're attempting to measure or get at, is the amount of kinetic energy of the molecules, okay, within that substance. So every solid, liquid, gas, unless it's at 0 Kelvin, the molecules are moving around a little bit. The molecules in this are moving around a little bit. The molecules in my coffee, they're moving around more than that, all right? It's warmer, it has a higher temperature. Cool so far? So the point here is that all of these special characteristics of water that we're talking about have to do with the fact that the molecule is so polar, okay? And in order to talk about why water is so polar, we had to talk about what's an atom and what's electronegativity. So all of that was really so we could talk about polarity. Great. Also, evaporative cooling is a thing that biological organisms take advantage of. You sweat and you let the liquid evaporate off of your body, and that cools your body as those molecules take energy with them. Evaporative cooling is a little more complicated than this because it also has to do with phase changes and phase change energy or state change energy, but we're not going to really talk about that. You should have talked about that, I hope, in chemistry, or maybe in, I don't know why, maybe you talk about it in physics. It's really cool stuff, you know, but I'm going to resist the urge to talk about it. This is going to be the first semester where I don't fall a week behind. It's going to happen, all right? Phases of water, all right? Like anything, water can exist in a gaseous form, a liquid form, or a solid form, okay? If you get any gas, remove a bunch of energy from it, it will transition into a liquid form. If you remove even more energy from it, it transitions into a solid form, okay? And the solid form occurs because the intermolecular interactions are very, very stable and the molecules aren't moving around a lot. If you heat up a solid, it melts. The molecules start to move around more. There's so much kinetic energy to the molecules that their intermolecular bonds are no longer stable. So that's what's happening with water. You've got an ice cube, you put energy into that ice cube, and the molecules start to move around, okay, as it raises in temperature, all right? As you put that heat energy and they move around. Eventually they're moving around where they're constantly moving. And then you put more energy into it and you get evaporation, and the things start to fly off. Now, one of the important characteristics of water is that its solid phase is less dense than its liquid phase. Therefore, the solid floats on top of the liquid. And we're in a biology class, so we tie this to biology, and it's actually incredibly important that this happens. All right? One example of why this is so important is lake ecosystems. Okay? In a lake, bless you, in the winter, in a lake, the lake freezes over, right? I don't know, I grew up in Washington State, and we used to go walk on the ice, and then inevitably you'd go too far out and you'd fall in, and it just was like a yearly thing, all right? There's liquid under there. If our lakes were made of iron, all right, and it was getting cold, the iron would solidify, and it would become more dense as it gets more stable, which makes sense, right? Because the molecules are moving around, and now they fall into place and they lock in with one another. But because of water's shape, all right, when it forms these stable hydrogen bonds, it makes this sort of hexagonal pattern, and it actually becomes less dense, all right? So a lake of molten iron, the things as they solidify would fall to the bottom. And over time, the entire lake of iron would freeze. But in a lake made of water, the surface layer freezes, and that surface layer continues to float. Eventually, it covers the entire lake, and this actually insulates, all right, the liquid phase of the water underneath. So lakes tend to not freeze all the way to the bottom, and that means that life can persist through those colder months. So it's actually very important that ice is a solid to explain ecology of lakes and things like that. Cool. It all has to do with the hydrogen bonds, which have to do with how polar the molecule is. Water, the universal solvent. It's called the universal solvent. Not everything is soluble in water, so it's a misnomer, right? But water is a very good solvent because the molecule is so polar. It's got regions of positive and regions of negative charge. So some things are super soluble in water, like polar molecules or ions. So right here, if you drop sodium chloride into a little beaker, all right, it can dissociate into its constituent ions. And because water's polar, water can interact with both the positively charged cation and the negatively charged anion. The positively charged or the partially positively charged portions of the water molecule, the hydrogen regions, will interact with the negatively charged ion, and the partially positive charged pole of the water molecule, or sorry, partially negative charged pole of the water molecule, the oxygen side, will interact with the positive ions. All right? Cool. I'm going to tell you right now, because everybody seems to miss this question on exam 1. I've got a question on exam 1 where I basically show you this. I remove chlorine. I tell you, hey, this particle's negatively charged. What's this? All right? That's oxygen. This is hydrogen. Oxygen's electronegative. Oxygen is the partially negative charged end of the water molecule. Fair enough? That's a freebie. I think if I hit escape, it'll just unzoom. No big deal. All right, cool. Thank you, thank you, thank you. So other molecules that aren't polar, they don't dissolve in water very well. Non-polar molecules won't dissolve in water very well. We'll talk about this in a second. Now, some proteins, sugars, things that maybe aren't, that won't dissociate, that won't come apart, all right, in water, they can still be held in solution because these big macromolecules like carbohydrates and proteins, they tend to have regions of exposed charge. So we look at this big blob of protein, all right, right here. It's got some regions that have partially negative charges and some regions that have partially positive charges on its surface. We'll talk about why when we get to biological macromolecules. But that means that water can interact with the surface of this protein, and so a little protein can float up and be soluble in water. All right? So many things are soluble in water because many things have some degree of charge on their surfaces. Water sticks to surfaces because those surfaces like even glass, all right, have some degree of charge to them, so the polar molecules can interact. Water doesn't stick to things that are non-polar, all right? So this is why if you've got maybe you've used a plastic graduated cylinder before, they always feel cheaper and they get discolored really quickly. And I mean, I don't think anybody likes them. It doesn't feel as nice to be dealing with a plastic graduated cylinder. But if you look at a plastic graduated cylinder next to a glass graduated cylinder, the meniscus is much deeper in a glass graduated cylinder than in a plastic graduated cylinder, where the surface of the fluid will look more flat. That's because a plastic is oil, all right? Plastics are made of petroleum. Plastics are just solid, hard oil, all right? And oil is a nonpolar molecule. We're going to talk about the lipids later, but it's a nonpolar molecule, so water can't interact with it. So you say oil and water can't mix, all right? And lipids, or the fats, okay, they won't mix with water because they're made up entirely of primarily of nonpolar covalent bonds. And remember, if you have nonpolar covalent bonds inside a molecule, the molecule itself is nonpolar, and like dissolves like. So a polar solution like water, you dump oil in it and it'll just sit in a layer. You shake it up and it dissociates into these little bubbles, and then those all just join together if you leave it overnight. Cool stuff? Now, amphipathic molecules. These things make oils soluble in water. They make nonpolar substances soluble in water. Amphipathic molecules are incredibly important. And what makes a molecule amphipathic? Well, it's got a region that is polar and a region that is nonpolar. All right? That's what an amphipathic molecule is. So the nonpolar region can associate with nonpolar solutions, and the polar region can associate with polar regions. So we're going to talk about phospholipids in the next slide, and then for an entire week as we talk about cell structure and function. Phospholipids are amphipathic molecules, and without them, your cells have no structure, okay? They're hugely important. So, what I'm hoping that I've gotten across to you so far, like dissolves like. Water's polar. Nonpolar things won't dissolve into water. Cool?
And amphipathic molecules make nonpolar solutions soluble in polar solutions.
Exactly, because what they can do is they sort of bridge the gap. And I'll repeat that. Amphipathic molecules make nonpolar solutions soluble in polar solutions. And actually, vice versa, it kind of depends on if it's let's say you're mixing oil and water, and if it's mostly oil, you're making the water soluble in the oil. You know what I mean? That's a terminology thing. It's annoying. Yeah, I don't need to worry about that. But here, let's take a look at what I'm talking about with the phospholipid. Definitely, for Bio 105, the most important amphipathic molecule, okay? So check this phospholipid out right here. It's got this long chain of carbon and hydrogen. And if you remember from last week, I told you that carbon and hydrogen form a nonpolar covalent bond, all right? That's one of the things that you have to just kind of remember, because the periodic table doesn't make it immediately apparent. It looks like it should be a polar bond or something, but it's not, okay? So you've got this long tail of carbon and hydrogen. This is made up of nonpolar covalent bonds. Therefore, this region of the molecule is all nonpolar. Then you've got this polar head region, okay, that actually has charge to it. You can see this oxygen has a negative charge, this nitrogen has a positive charge. It's very polar up here. The atoms involved in the bonds here, they differ greatly in their electronegativity. So you've got this phospho or phosphate group here attached to this nitrogen-containing molecule. You're not going to be quizzed extensively on the structure of a phospholipid beyond knowing, okay, that it has a polar region and a nonpolar region, making it amphipathic. And now, polar things are hydrophilic, nonpolar things are hydrophobic. So a polar molecule is hydrophilic because it will dissolve in water, or water can associate with it. Water-loving. A nonpolar molecule is hydrophobic, all right? Water-fearing. It's nonpolar, so it's not soluble, okay? And these amphipathic molecules, they have hydrophilic regions and hydrophobic regions. Cool so far? Awesome. So what happens if you just take a bunch of phospholipids and you dump them into some water? Well, the tails will all eventually just sort of auto-assemble and join up together to make this little hydrophobic region. Okay, why does this happen? Because the hydrophobic regions of the molecule are all repelled by the water molecules. And so this is a more stable energetic state to have all of the hydrophobic tails associated together. If one of these molecules starts to flip and come out of here, the water molecule will just be like, nope, get back in there, get back in there, okay? So this is one of the structures that just spontaneously assembles if you drop phospholipids into some water. All the hydrophilic heads just face outward and it makes a bubble. Also, you will get these phospholipid bilayer sheets, and these are so important. You'll never get quizzed on a micelle. You did probably make soap in a chemistry lab at some point, or you will probably make soap in a chemistry lab at some point. And they use this same terminology with soaps. What's a soap? Basically, you carry out a process of saponification, okay, on a lipid, and you turn it from a hydrophobic nonpolar molecule into an amphipathic molecule. And now imagine that in the center of this micelle right here, there's a little blob of oil. It would be suspended with these hydrophobic tails, all right? And then it's now in suspension, and it could be washed away. That's how soap works on your skin. It's an amphipathic molecule. You put it on your skin. It interacts with the oil sheet on your body, all right? And then the hydrophilic heads are facing out, and when you take water and wash it off of your body, that pulls away the oils, okay? So that's what soap is. It's amphipathic molecules. And you all still watch Fight Club, right? Because you're in college, you have to watch Fight Club. Remember when they go to the liposuction clinic and they steal all the fat and they're going to make the soap with it, okay? That's why that makes sense, is because you're taking a bunch of fat molecules and you're going to run them through a chemical reaction and you're going to turn them into amphipathic molecules, right? And then the bag rips, and doesn't it like dump all over him or something that's really gross? Is that what happens in that movie? Yeah, I think so. All right. I don't know. Phospholipid bilayer, all right? This here, that micelle stuff, that's all just for your interest. Phospholipid bilayers are really, really important in biological systems. This is the foundational structure of all of your cell membranes. This is the basic boundary that separates the inside of an organelle from the outside of an organelle. This is what makes cells function, okay, is setting up this phospholipid bilayer. All righty? Now, check it out. Same sort of interactions. This is in an aqueous solution, so it's a polar solution. The hydrophilic heads all face out and interact with the water, and all the tails get sort of pushed into proximity. So you get this domain of hydrophobicity, which is what I always call it. I saw it in a textbook a long time ago, and sometimes they call it the hydrophobic region or the hydrophobic domain, but I think that domain of hydrophobicity is just way cooler. So check this out, right? You've got the domain here, and it becomes a semi-permeable barrier. Think about this. Anything soluble in the water out here or soluble in the water over here, okay, is polar. That means that it's not going to readily dissolve into this hydrophobic domain. So this hydrophobic domain in the middle of a phospholipid bilayer prevents free movement of molecules into or out of a cell. Question, please.
So in theory, you could do like just plain like fresh water on the bottom, form a phospholipid bilayer, and then pour salt water on top, and they wouldn't mix?
Well, they would not, yeah. The salt water, it would dissociate over here, right? It becomes sodium and chloride ions, and then it would not mix into here, like at all. And that's actually great, because when we talk later about osmosis, that's what goes on with osmosis. Ions absolutely won't cross here. Points for you. No problem, that's great. Yeah, so things, that's the idea. That's actually really good. I'm gonna give you 7 points. Yeah. So if I pour, you're great, write them down. They expire at the end of the day though. So you put the salt in here, right? It dissociates into ions, and remember, ions are like super hydrophilic. They're super polar. That's the way that I was telling you to think about them for our rules of solubility. Those ions are definitely not crossing this domain of hydrophobicity because this is all nonpolar and they are super soluble in the water. It doesn't make sense that they would cross. Cool? Awesome. I know we spent a lot of time on this slide, but trust me, phospholipid bilayers, we're going to come back to them again and again and again, and hopefully the groundwork we laid today will serve you well in the future in making the more complex concepts more approachable. Summing up water's properties. Water sticks to things, water sticks to itself, water changes temperature slowly with its high specific heat, water can dissolve a lot of things. It all comes back to the polar nature of the water molecule, which allows it to form these hydrogen bonds. Any questions about water? Is water a polar molecule or a nonpolar molecule? That's right. That's right. Cool. I don't want anybody to miss any questions about that. Awesome. pH. Time to talk about it. All right, let's talk about the pH scale. You know about things being acidic or things being basic and how like, oh, I don't want to get a bunch of battery acid on my skin. That's bad. Everybody's always whining to me like, John, why are you pouring this battery acid on my foot? But you know what? I'm trying to teach them a lesson about the pH scale, but they're always too busy, all right? So let's talk about it. You've heard about the pH scale. You know about things being low pH, being acidic, and high pH being basic. Let's talk about what it actually represents, okay? The pH scale is the inverse log of the concentration of hydrogen ions in a solution. That doesn't make a ton of sense, all right? Neutral pH is 7. A neutral solution has a pH of 7. Okay, so your blood has a neutral pH, all right? Now, very extreme pHs, okay, represent harmful substances for biological processes, like bleach, or a battery acid, or lemon juice, okay? So check this out. I am gonna sort of just lay the groundwork and then we'll talk about what the inverse log of the concentration of hydrogen ions means. At a very low pH, you've got a bunch of positively charged hydrogen ions in a solution, okay, in an aqueous solution. At a very low pH, at a very high pH, did I say low for this? I meant to. Okay, cool. At a low pH, high concentration of hydrogen ions. At a high pH, low concentration of hydrogen ions. So water sort of exists in this weird, water's even in pure water, water's doing stuff, all right? It's dissociating into hydrogen and hydroxide ions. That hydrogen ion forms hydronium sometimes, which is H3O. It's all wild. And then water does all these crazy things at its actual intersection with some other substance or with gases. You could teach an entire class on water, okay, and just what's actually going on with it. It's really complicated. So I'm going to keep it as simple as possible without sacrificing the understanding. Let's see here. Boom, boom, boom. Oh no. All right. Document camera. Zoom it out. Okay, pH. Inverse log of concentration of hydrogen ions. These square brackets, they always mean concentration. Get used to using them and seeing them because they don't go away. People just always use these square brackets for concentration. So somebody's talking about concentration of glucose, they write the word glucose and put it in square brackets. All right? Inverse log of hydrogen concentration in moles per liter, which remember is a molar solution. So what does this mean? A pH of 3 equals 10 to the negative 3 molar hydrogen ion concentration. Okay, look, it's the inverse log. So it's just this exponent with the negative removed. Now, what does that actually mean? Remember, that means that's 1 times 10 to the negative 3 moles of hydrogen per liter. All right? Or, remember, 1.0, if you're going 1 times 10 to the negative 3, you just go 1. Does that fix it? Cool. 3. 1, 2, 3. 0.001 moles per liter. All right? Cool so far? Now, check this out. I already told you that at a low or more acidic pH, there's more hydrogen ions. Now let's look at what I actually mean by that. Let's zoom it out a little bit maybe. How's that look? Okay, cool. pH of 10 equals 10 to the negative 10 moles hydrogen ions, okay? Which is 1 times 10 to the negative 10 moles of hydrogen ions per liter, okay? Which is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 0.00000000001, okay? So do you see here why a high pH has a lower, this concentration here, this number right here is smaller than this number. I think we can all accept that. So a higher pH means a lower concentration of hydrogen ions. Cool? All right. Now here's sort of the last weird thing here, is that in an aqueous solution, the concentration of hydrogen ions times the concentration of hydroxide ions will always equal 10 to the negative 14. They're in sort of a balance. So if the pH equals 3, the pOH equals 11. Now, do we remember when you're multiplying numbers that have exponents, you add them together? So a pH of 3 would be 10 to the negative 3. A pOH, or the concentration of hydroxide ions, would be 10 to the negative 11. 10 times negative 3 times 10 to the negative 11 equals 10 to the negative 14. Cool? Yeah, I'm pretty stoked on that sheet of paper. That's pretty good notes. I like it. If you want to take a picture of it later, you can. I don't know. I've got it right up here. You feel free. Let's go ahead and turn that light off because it's annoying. Okay. So why is a pH of 7 a neutral pH? Because at that point, we've got the same concentration of hydrogen ions as hydroxide ions. So the net charge of the solution, all right, is neutral because it's the same concentration of positively charged hydrogen ions and negatively charged hydroxide ions, all right? pH of 7 would mean concentration of hydrogen ions is 10 to the negative 7. 7 plus what equals 14? 7. So the concentration of hydroxide ions would be 10 to the negative 7. They're the same. How do we feel about this? Cool. I already answered this question, so we're just going to keep on keeping on. And yeah, great. What is the pH of a solution with a hydroxyl or hydroxide ion concentration of 10 to the negative 12? Now, hydroxyl versus hydroxide, I still see them both being used interchangeably. It's pretty weird, but let's not worry about that too much, okay? If it's 10 to the negative 12 molar for the hydroxide ion concentration, yeah? A, solid, pH of 2. You just take this number, subtract it from 14. Or add it to 14 technically because it's a negative number. But yeah, we cool with all that? If something's an acid, if you put an acid into a solution, it pumps in a bunch of hydrogen ions. If something is a base, it removes a bunch of hydrogen ions. All right? And this generates more hydroxide ions. Cool. Now, why does any of this matter? Well, we've got all these big molecules, okay? And if you've got a bunch of charged particles interacting with these big molecules, they mess up the interactions within the molecule that hold those macromolecules together. I'll bring this up again when we talk about the biological macromolecules, specifically when we talk about protein structures, but just very briefly, imagine that there's a long protein chain here, and this elbow is negatively charged, and my hand is positively charged. Naturally, the protein will take on this conformation, okay? It takes on this shape, all right? But if you dump a bunch of hydrogen ions in here, well, then those hydrogen ions are gonna just associate with the positively charged region over here, and now the protein no longer has its form, okay? And with these biological macromolecules, form is function. Cool. Promise we'll talk about it later. But I just wanted to mention, like, why is an acid or an extreme base harmful? Usually because it messes up the structures of biological macromolecules. Moving forward, buffers, all right? Buffers, they can absorb hydrogen ions, they can release them, so they maintain pHs within certain ranges. So your blood has buffers in it, okay, that keep your body, that keep the pH in the blood close to neutral. And if you're doing experiments in the lab, they'll have with pH, or you need to make solutions have a certain pH to foster the growth of certain organisms. Then you have all these different buffers that sort of maintain pH near a certain level. So there are buffers that maintain pH near 4 and buffers that maintain pH near 10, okay? There are different types of buffers. We're not going to worry too much about it because it's not chemistry class. You just need to know that they exist. Cool? Awesome. About 8 minutes left. Okay, no, we're doing great. Let's crush this. Seen it, moving forward. Learning objectives, check it out, moving forward. All right? Form is function. Look, it's like I knew this was coming. It's like I've taught this class before or something. The structure of molecules dictates their function, okay, to a large degree. You already know that if water didn't happen to have the shape that it does, it wouldn't be a polar molecule, okay? So, shape of molecules matters. Shape of big molecules also matters. A lot of drugs work by mimicking the shape of some natural neurotransmitter or something that your body normally responds to. If you mimic the shape of it, then you can get it to respond in the same way. This whole slide exists to just say shape matters of these molecules. Okay? Now, carbon, it can do a bunch of versatile bonding. So molecules that are based around a carbon skeleton can have a lot of diverse shapes. So if you're gonna base life around some skeletal molecule, carbon makes sense. It can form the maximum number of covalent bonds, which is 4, okay? And yeah, it can participate in double bonds. It can make all sorts of things. The bonding versatility lets it make a bunch of diverse molecules. Cool. All right, let's not worry about this. It's covalent bonds. It's most likely to form covalent bonds. Okay, we have to talk about, since we're talking about the importance of the shape of molecules, we need to talk about isomers. So we've talked about isotopes already, okay? But now we're talking about isomers. So an isomer is a molecule, okay? Or let's say you got a molecule with a certain number of different atoms in it. But the bonding pattern, and therefore the shape of the molecule, could be different. Just because you have a certain number of each atom doesn't mean that you're going to have the same shape. So right here, butane and isobutane. Each of them have 4 carbons and 10 hydrogens. But they are arranged slightly differently, so they have a different form, so they behave slightly differently. Okay? So we've got structural isomers, geometric isomers, and enantiomers. Okay? The structural isomers are the most straightforward. They differ in spatial arrangement of atoms. All right? Covalent bonds of atoms. Now, that is true of all of the different isomers. They differ in their sort of arrangement, but the geometric isomers and the enantiomers are specific. So the structural isomers are like your baseline isomer, okay? There's some different arrangement of the covalent bonds within the molecule. Your geometric isomers, which are also called the cis-trans isomers, okay, because those are the names given for the different isomeric forms, okay? These are all based around double bonds. So if a molecule has a double bond in it, then there can be cis forms of it and trans forms of it. These are different isomers. So let's use my hands. I use my hands and my arms a lot. They're good models of this stuff. So check it out, right? You've got a double bond. Let's start with this. I'm making a big molecule now, okay? There's a bunch of bonds in here. And let's say that the bond here, formed by my thumbs, is a single bond, okay? If it's a single bond, then the big molecule can rotate around that and take on all these different conformations. Therefore, this is the same as this, is the same as this, because when it's in a solution, it can be all those different forms, okay? But if you have a double bond in a molecule, so 2 electrons being shared between shells of carbon atoms, or it's not always carbon, but if you have a double bond in a molecule where 2 electrons are being shared, the bond is much more stable, and therefore the molecule can't freely rotate around it. So that means that this molecule here is actually different from this molecule right here, because it can't freely shift between the 2 states. Does that sort of make sense for folks? So that means that if you got a double bond, it matters, okay? Even though the bonding pattern is still the same, right? Like if each one of my fingers is a hydrogen, for example, it's still carbon to hydrogens, but these are different, okay? You can take a look, I guess, here on the actual slide as well. So, cis means same, trans means opposite as a prefix here. So in a cis isomer, the opposite sides of the double bond are mirror images of one another. They're the same, okay? In a trans isomer, the opposite sides of the molecule across the double bond are opposite, all right? They're flipped. Cool? Awesome. So check it out. Draw a line through the double bond, and if it's a mirror image on either side, you're looking at a cis isomer. If it's not a mirror image on either side, you're looking at a trans isomer. These are geometric isomers. There it is. Double bond matters because it's so stable. The last one is the enantiomer, okay? And enantiomers are weird because they have the same arrangement here, same sort of chemical structure, but they're mirror images of one another. And the only way to convey this is with the hand example, okay? Because this is what every class does. My right hand is a mirror image of my left hand. They are perfectly identical. I've spent thousands of dollars on surgery to make them exactly identical. Because, if they don't look the same, then I don't know, it would be a lightning strike or something. All right? But look, they're mirror images of one another, but a glove for my right hand would not fit on my left hand, assuming that it's like a driving glove that has a shape to it. In this situation, I'm Ryan Gosling in Drive. All right. And in every situation, I'm Ryan Gosling in Drive. Let's just establish that right now. Look, my hands don't lay over one another perfectly, okay? They can't take up the same space. They are enantiomers of one another. They're different, even though it looks like you could just rotate this and it would be the same. They're different. And then this is a meme. Here we go. Look, it's Samuel L. Jackson and Samuel D. Jackson, because the L and the D are the different isomer abbreviations, and these mirror images of himself. See, L isomer, D isomer. And that is going to be where we have to end today, unfortunately. Okay? We've got just a few more slides here. We'll get through this no problem on Wednesday, get into our biological macromolecule stuff. Thank you all for coming to class. It's always a pleasure. Have a good rest of your day. And yeah, I'll see you on Wednesday. Goodbye, recording people.